Welcome to today's Explainer. I am absolutely thrilled you're here because we are about to go on an incredible journey right into the center of our own cells. We're going to explore a microscopic world that is so highly organized, so densely packed, and honestly, so incredibly dynamic, it makes the logistics of a bustling metropolis look like a piece of cake. We are talking about the microscopic geography of the nucleus. Just think about this for a second. Every single cell in your body contains roughly 2 meters of DNA. 2 meters! So how in the world do 2 meters of genetic material fit into a microscopic nucleus without tangling into total, unmanageable chaos? If you pose this mind-bending spatial problem to most people, they might think the DNA is just, shoved in there randomly like a bowl of spaghetti. But that couldn't be further from the truth. The nucleus isn't some messy soup, it's a meticulously organized map.
And that brings us to our core concept for today, subnuclear compartmentalization. Basically, this is the dynamic spatial organization of the genome relative to specialized membraneless structures inside the nucleus. Imagine a massive, bustling city. You wouldn't put a loud, heavily polluting industrial factory right next to a quiet suburban neighborhood, right? The city is zoned into specific districts. Well, the nucleus does the exact same thing. It zones our genome into distinct neighborhoods where genes are either heavily guarded and silenced or actively transcribed and processed.
Okay, let's dive into this. Here is our zero-to-hero roadmap for today. We're going to quickly map out the geography of the nucleus, meet the architects, like XIST and Mallet1, and check out the cutting-edge tools scientists use, specifically epitopes and the TN5 enzyme. Then, we'll see how they map it all with RT&Tag, add the dimension of time with SLAM-RT&Tag, and finally, look at the master blueprint of why this all matters for our health.
So, Part 1. The geography of the nucleus, specifically the active versus silent zones.
To really understand how our genes are regulated, we have to recognize that this structural geography dictates everything. Location literally dictates function. When we look at a stained nucleus, we can see the DNA everywhere. But we also see these striking, brightly glowing green clusters marked by a specific antibody. Those glowing dots are called nuclear speckles. These are the active neighborhoods. They are highly concentrated, membraneless bodies packed with RNA processing machinery, specifically splicing factors. When a gene is actively transcribed, its RNA often migrates right into these bustling speckle centers to be processed.
But the nucleus isn't all hustle and bustle. In stark contrast, we have other areas, often marked in red when scientists stain them, that represent something entirely different: polycomb domains. If nuclear speckles are the bustling factories, polycomb domains are the highly secure, silent vaults. These are heavily condensed, repressive territories where genes are strictly switched off. So we have this incredible contrast right inside the same space: the active speckles and the silent polycomb domains. Moving to Part 2, the RNA architects XIST and MALAT1. So who or what actually builds and maintains these vastly different nuclear domains? Enter the master architects, long non-coding RNAs. Now, these aren't your typical RNAs that just go off to make proteins. Their very existence acts as a physical structural scaffold for the genome. On one side, we have XIST. XIST is the architect of those silent polycomb domains. It's a stable resident that essentially coats and silences chromatin. Most famously, it orchestrates the complete shutdown of the inactive X chromosome in female mammals. On the flip side, we have MALAT1, which is the architect of the nuclear speckles. Unlike most transcripts, MALAT1 lacks a standard poly-A tail. Instead, it just hangs out in the speckles, acting as a dynamic scaffold to organize those active splicing factors. 2 RNAs, 2 entirely different architectural jobs.
Part 3. Let's talk about the precision tools scientists use to actually see this: epitopes and TN5.
For decades, mapping exactly which RNAs were in which of these microscopic domains was nearly impossible. Breaking the cell open to study it would just mix everything into a giant molecular soup. To map this geography in situ, meaning right there in its natural place, scientists needed a way to essentially drop a pin on the map. That pin is the epitope. An epitope is simply the exact molecular address on a protein target that a specialized antibody binds to. By using an antibody that recognizes an epitope unique to a nuclear speckle, or maybe one unique to a polycomb domain, researchers can send a targeted molecular GPS directly to that specific subnuclear zip code.
And once their antibody finds the right address, we need a tool to do the heavy lifting. Enter the TN5 enzyme. This is an absolutely fascinating piece of biological machinery. It's a hyperactive enzyme originally found in a bacterium with the rather unfortunate name Vibrio diarrheacus. But what makes TN5 so special is that it's a biological multi-tool. It can simultaneously chop up nucleic acids and tag them with specific DNA barcodes in a single, elegant step. We call this process tagmentation.
Which brings us nicely to part 4, mapping this spatial GPS with a technique called RT&Tag. When we combine the GPS targeting of an antibody with the barcoding power of that TN5 enzyme, we get a revolutionary technique for mapping the transcriptome spatially, RT&Tag. The workflow is brilliant. First, we permeabilize intact native nuclei. We don't destroy them, we just poke tiny holes so our tools can slip inside. Second, we send in our primary antibodies to bind to our target subnuclear epitope. Third, we introduce the TN5 enzyme, which is fused to a special adapter, along with a primer. That adapter physically tethers the TN5 directly to the antibody. Finally, we reverse transcribe the RNA located right there in that specific neighborhood, and our tethered TN5 immediately chops it up and barcodes it.
What's really incredible here is how the antibody physically anchors that TN5 enzyme directly inside the RNA's microscopic living room. Because this all happens in situ, we only sequence the RNA that is physically touching or immediately surrounding our target compartment. We aren't blending the whole cell into a smoothie anymore. We are meticulously surveying specific intact neighborhoods at incredibly high resolution.
Okay, part 5, adding the dimension of time with SLAM-RT&Tag. Now, a 3D map of these neighborhoods is a huge breakthrough. But biology isn't static. RNAs are constantly being born, moving around, functioning, and degrading. By turning our 3D map into a 4D kinetic tracking system, researchers created SLAM-RT&Tag. They start by feeding the cell a labeled molecule called S4U. As the cell makes new RNA, it incorporates this S4U, leaving a literal kinetic breadcrumb trail. Next, they treat the cells with a chemical that modifies those specific breadcrumbs. When we run our RT&Tag process, the reverse transcriptase enzyme gets confused by the modifications and misreads them, causing a highly specific T to C conversion in our genetic data. Finally, using a software brilliantly named SlamDunk, you gotta love scientists naming things, we can quantify exactly how old an RNA molecule is based on these conversions. By tracking the flow of RNA over time, scientists made a massive discovery about those nuclear speckles. They found that speckles act as quality control checkpoints that temporarily store incompletely spliced transcripts until the splicing is actually completed. Speckles aren't just passive storage closets. They are active, highly dynamic security checkpoints, ensuring only fully processed, mature RNAs are allowed to exit the nucleus. And the time data backs this up perfectly. SLAM-RT&Tag revealed that transcripts born adjacent to the silent polycomb domains are extremely long and transient. They are synthesized and then they move away incredibly fast. But in stark contrast, transcripts inside the nuclear speckles experience deliberate traffic jams. They linger there with a surprisingly long half-life of 2 to 3 hours, while the splicing machinery meticulously pieces them together.
Finally, part 6, the master blueprint, and why this matters for health and disease. So we've mapped the geography, met the RNA architects, and tracked the genetic traffic in 4D. But why does this microscopic biological cartography actually matter to you and me? Well, it matters because the breakdown of these spatial neighborhoods is a root cause of severe human disease. When those polycomb domains become misregulated, they can mistakenly silence homeostatic genes that our bodies desperately need to survive, and that directly drives tissue decline and the physical realities of aging. Meanwhile, if RNA architects like MALAT1 are overexpressed, or if nuclear speckles just become disorganized, the resulting chaos in RNA splicing is a hallmark of many human cancers. The structural integrity of the nucleus is, quite literally, the structural integrity of our health. Which leaves us with a truly mind-blowing question to chew on. We now have the tools, like SLAM-RT&Tag and the TN5 enzyme, to map the exact 4D coordinates of disease-causing RNAs. We know exactly which subnuclear neighborhoods they are hiding in. So, if we can map them, could we one day deliver targeted spatial therapeutics straight to a single subnuclear zip code? Could we cure diseases at the architectural level of the cell? It is a thrilling frontier of medicine, and it all starts with understanding the microscopic geography of the nucleus. Thanks so much for joining me on this incredible deep dive. I can't wait to explore the next complex topic with you right here on The Explainer.